Welcome to the Downeast Institute's sixth and final climate change seminar of 2022. Thanks for joining us. My name is Sarah Randall, and I'm the Associate Director of the Downeast Institute. This seminar is part of our series of Downeast discussions where we share some of the groundbreaking research being conducted through Downeast Institute at our facility in Beals, Maine, and also in the field around the state of Maine. This summer, we've held these seminars every week during the lunch hour, and the seminars primarily focus on how commercially important shellfish are being impacted by climate change and how to adapt to the changing marine ecosystem. For those of you that don't know, Downeast Institute is a nonprofit marine research laboratory, science education center, and shellfish production and research hatchery located in the heart of Downeast, Maine and Washington County on Beals Island. We also serve as the Marine Science Field Station for the University of Maine at Machias. So today's seminar will take about 45 minutes. It's scheduled to end at 1 p.m. So please, this is a regular Zoom meeting. It's not a webinar format. So please keep yourself on mute during the presentation. And after the presentation's over, we'll take as many questions as possible. And you can ask a question by raising your hand on the Zoom or writing it in the chat. And the presentation is being recorded and will be uploaded to YouTube, just so you know. And I'd like to thank Hannah Green, who is our Island Institute Fellow, for helping today with the technology. And as she's helped with pretty much all our other climate change seminars. And it is one of the last few weeks of Hannah's time with us. And she spent 2 years at Downeast Institute working on community outreach and education and data collection. And so I just wanted to thank her for her time and her efforts in the community of Beals. And she's really done a lot. So I just want to take the time to thank Hannah for her contributions. Today, we are going to hear from Downeast Institute 2022 Fellow Ray Czaja. And he is going to discuss using meta-analysis to explore the roles of upwelling exposure and experimental design decisions in bivalve ocean acidification studies. So Ray, in addition to being a 2022 Downeast Institute Fellow, is entering his 4th year of PhD study at Stony Brook University. While most of his research pertains to climate change impacts on the Atlantic surf clam fishery, this particular study presentation that he's about to give today is about many other bivalves. Ray approaches range from controlled lab experiments to modeling techniques, and he aims to assess both individual and population level responses. He doesn't discriminate among climate change phenomena and studies impacts of warming, acidification, food availability changes, and wind pattern changes. This presentation is very exciting because he has yet to show any of these results to anyone. So we get to see what he has learned. And with that, I will turn it over to Ray for his presentation.
Thank you, Sarah, for that introduction. You covered a lot of what I was going to say, so I'm probably going to repeat myself here for a few moments. So hi, everyone. As Sarah mentioned, my name is Ray Czaja. I'm a PhD student in the Marine Animal Disease Lab at the School of Marine and Atmospheric Sciences at Stony Brook University. I am also one of 2 graduate student fellows here at DEI this summer. And so as Sarah mentioned, most of my work pertains to climate change impacts on the surf clam fishery. However, today I'm going to be talking about a few bigger picture questions. More specifically, we're going to look at bivalves from all different species, all different locations, all over the world, all different populations. But we're going to focus on ocean acidification. More specifically, we're going to talk about the roles of experimental design decisions, as well as potential local adaptation via mechanisms such as upwelling exposure and variability of ocean acidification studies. And so here's how today's presentation is going to go. First, a very brief and familiar and elementary background on OA, as well as OA and bivalves. I'm then going to highlight some gaps in the OA bivalve literature, more specifically some questions that have yet to be answered regarding the roles of decisions that scientists make, as well as potential local adaptation. I am then going to talk about how meta-analysis can be used to address some of these questions. I'm going to assume that most of us in the audience are not too familiar with the meta-analysis. So we're going to define it, talk about what it is, how it works, and how it can be used to address these literature gaps. Then I'm going to point out my specific study objectives, and very briefly, the methods and data analyses that I conducted, and then the fun part, the results, and then the discussion. And so right now, or as Sarah mentioned, this is my first time presenting this material to an audience. And I'm pretty excited because I'm also in the middle of writing the paper for this study, or perhaps a more accurate statement would be I'm in the middle of some writer's block of my discussion. So I'm hoping that you folks in the audience today may provide some questions, feedback, or comments that'll help me get over that roadblock hurdle. So first, that very brief intro on ocean acidification. We have anthropogenic activity releasing all this carbon dioxide into the atmosphere, right? And so a good portion of that hangs out in the atmosphere, contributes to the greenhouse effect, and leads to ocean warming. However, a good portion of that CO2 also gets directly absorbed into the ocean. And when it does that, it sets off a chain reaction in the carbonate chemistry system of the ocean, often referred to as ocean acidification, or OA. And three of the big symptoms of OA are an increase in PCO2, a decrease in pH, and a decrease in saturation state. And bivalves may be particularly vulnerable to OA due to their calcifying shells. And so bivalves may respond to OA all different ways, all different responses we can measure here. One of the big ones is, of course, survival. Sometimes ocean acidification can be so stressful that it can lead to increased mortality or decreased survival for bivalves. However, another fisheries relevant response is growth. Sometimes bivalves decrease growth in response to OA due to the stress associated with this abiotic condition. And now one of the two responses that I'm interested in is respiration rate, or I abbreviate it as RR. And I view respiration rate as one side of this two-sided metabolic or energetic coin, where this coin leads to, again, changes in growth. Respiration rate is directly tied to growth. And respiration rate is often quantified as oxygen consumption. But I also like to think of respiration rate as the amount of energy that's used for maintenance, and therefore dictates the amount of energy that can be used for growth. So I like looking at respiration rate because I think it provides insight into growth changes and metabolic activity changes. And on the other side of the energetic or metabolic coin is feeding rate. But for bivalves, we often say clearance rate because they're clearing particles, often phytoplankton, from the water column. And so clearance rate, just like respiration rate, may change in response to OA. And clearance rate, just like respiration rate, also affects growth. The amount of energy taken in via feeding obviously affects the amount of energy that can be used for growth. So what do we know about bivalve clearance rates and ocean acidification? Well, we know that different bivalves respond different ways. So for example, Cole et al. Found that their oyster larvae in the UK decreased clearance rate in response to OA. However, last summer here at DEI, we found that surf clam larvae did not change their clearance rate in response to OA. So then this begs the question of okay, why do different studies find different outcomes? Or maybe another way to phrase that is, why do different species or populations respond differently to ocean acidification? And maybe it comes down to the decisions that scientists make when designing their experiments. For example, maybe these two different studies had different exposure times. In other words, maybe we exposed our surf clams to OA conditions for longer amounts of time than Cole et al. Did. Maybe it has to do with different changes in carbonate chemistry parameters. So what do I mean by that? Well, for our surf clam larvae, we had two pH treatments, right? We had an ambient or control pH of 7.7, and then an acidified pH of 7.3. So we exposed our surf clams to a drop of 0.4 pH units. Perhaps Cole et al. Had a different approach there. Maybe they had an ambient pH of 8.0 and an acidified pH of 7.2. So their drop in pH would be 0.8 units. Maybe it's that different change in the drop in pH that affects the outcome and causes these differences. Maybe it's different temperatures too. For example, our surf clam larvae didn't get above 23 degrees Celsius. However, perhaps colloidal used warmer temperatures. And although temperature wasn't a factor of interest necessarily, maybe the temperature of the experiment did indeed affect the outcome. Or perhaps we scientists are not in as much control as we think we are. Maybe it's less about the decisions we make and more about some deep-rooted biological mechanisms. More specifically, maybe it's because different species or different populations are used to different environments. So for example, we may have intertidal and subtidal bivalves. Granted, these two experiments were conducted on larvae, so just bear with me here regarding the extrapolation to adults. So up here in Maine, surf clams can be found in the intertidal, but they're predominantly a subtidal species, whereas oysters very commonly live in the intertidal. And so intertidal species deal with a whole different suite of abiotic stressors than subtidal species. And so maybe because these animals are used to different abiotic stresses, they're going to be responding to the abiotic stress of ocean acidification differently. Perhaps it's not about intertidal or subtidal, perhaps it's about where in the water they are, maybe infaunal versus epifaunal. So for example, surf clams are infaunal animals, they live within the sediment. Oysters, epifaunal, on top of the sediment. As Rob was talking about a couple of weeks ago, that pore water sediment carbonate chemistry can be very harsh. And so infaunal organisms may have to deal with that harsh pore water chemistry, whereas epifaunal organisms not so much. And so maybe because these animals are exposed to pore water chemistry versus non-pore water chemistry, that's going to affect how they're going to respond to anthropogenic acidification. Or perhaps it has to do with whether or not the animals, the populations, the species are exposed to upwelling. So let's talk about upwelling for a moment. I'm going to define it for those who are unfamiliar with upwelling and talk about how upwelling is related to ocean acidification. So let's take an example stretch of coast here, right? We have a stretch of coast running from north to south, say, for example, California. And we have this alongshore wind component. We have wind going from the north to the south. Well, this alongshore wind component is going to result in what's called Ekman transport. And very simply put, you could think of Ekman transport as kind of a combination of the Coriolis effect and friction. So more specifically, this Ekman transport due to this wind is going to cause this near coast, near surface water to be pushed offshore. Water mass that was over here is now being pushed way offshore. And well, so the water level near the surface, near the coast, it's not going to drop, right? The water has to be replaced with water from elsewhere. So what happens is water from at depth is brought to the surface and brought to the coast to replace that water. More specifically, or rather you can say water at depth is upwelled over here. And so due to high respiration and high decomposition rates, this water mass is pretty low in dissolved oxygen, pH, and saturation state. So it's naturally acidified. So when you have upwelling, this naturally acidified water mass is going to be brought to the coast, to the surface, where we have all of this biomass and abundance of life. And so my hypothesis is that bivalves that live in these high upwelling areas, because they're used to these naturally acidified conditions, they may be more resistant, more resilient, or less sensitive to anthropogenic ocean acidification. Whereas if we have wind blowing the other direction to the north, and we're then going to have Ekman transport going to the coast as opposed to away from the coast, that's going to result in what we call downwelling rather than upwelling. But downwelling, we're not going to have these naturally acidified conditions near the coast. So these bivalves may be a little more sensitive to anthropogenic acidification because they're not used to that acidification like the upwelling bivalves may be. And so this idea of, these environmental differences, different upwelling exposures, this is kind of getting at the idea of local adaptation. In other words, species or populations of organisms from different areas may be locally adapted to certain stressors, and that may help determine how they're going to respond to future stressors. So these, broad questions regarding experimental design and environmental characteristics and local adaptation are interesting, but how do you answer them? You can't go ahead and perform dozens of lab experiments all over the world as one group of scientists. It's going to cost a reasonable amount of money. However, what you can do is you can analyze the data that's already there. you can gather the previously accomplished lab experiments and sort of reanalyze their data to address some of these hypotheses, to put values to some of these hypotheses and quantify some of these relationships. That's very simply put the idea of a meta-analysis. Let's talk about how a meta-analysis works and what it is. When I was first learning about meta-analysis, I read this phrase, it's like comparing apples to oranges. And people often use that phrase in sort of a negative light, right? You say you can't compare apples to oranges to make a point about, you can't group these things, you can't compare things. And oftentimes that phrase is used appropriately and is correct. But can you compare apples and oranges when your group of interest is all fruit? And that's kind of the idea of a meta-analysis. You have maybe clams as your grapes. You have mussels as your melons, and oysters as your oranges, and scallops as your pineapples. Sure, these are all different fruit. They're all different bivalve taxa. But if your group of interest is all bivalves together, you can try to group them and compare them to come to some more broad conclusions about all bivalves together. So what are the specific questions that a meta-analysis can ask, or answer rather? One thing a meta-analysis do is it can tell you, okay, when I group everything, what's the general consensus response? It's nice if you have all these different studies that provide insight, but what if you just want to be able to make a broad blanket statement? That's what a meta-analysis can help you do. So again, with a meta-analysis, you can group different species, different populations, habitats, parts of the world, and even life stages to come to that consensus response. Granted, you should account for these differences in your model, but nevertheless, you can still try to come to these broad conclusions. And something else a meta-analysis can do, which I think is the more cool aspect of a meta-analysis, is you can try to explain some of that between study variability. You can try to find support for these hypotheses regarding the roles of experimental design and local adaptation. You can figure out why different populations or species or studies come to different outcomes. Again, for example, our oyster larvae decreased clearance rate, though surf clam larvae did not. A meta-analysis can help you figure out why. And so to help explain how a meta-analysis works, like the structure of it, let's talk about the structure of a typical OA bivalve lab experiment. Just like you can perform here at DEI at our OA lab, you may have two pH groups, right? pH of 8.0, your control or ambient, and a pH of 7.6. And so to see differences between those two pHs, you would have multiple tanks with multiple scallops at each treatment level. And then you perform your study, you get your measurements, and then you're going to wind up with an individual value or an individual data point for each of the tanks, which are the experimental units. And the meta-analysis kind of works the same way, but instead of a tank being the value or the data point for the experimental unit, the individual study is the data point for the value for the experimental unit. And so, essentially your N winds up being the number of studies that you use in your meta-analysis. And that sounds great, right? But then the hard question is, okay, how do you put an individual value to a whole study that has all these values and numbers and conclusions associated with it? And it may be a bit simpler than you would suspect. More specifically, what you do is you calculate what's called an effect size for an individual study. An effect size is often referred to as standardized mean difference because it's sort of just that. It's the difference between the means of your treatment groups that's standardized so you can group and compare studies. So let's talk about how an effect size works in practice. So let's look at this study. Again, oysters, how oysters physiologically respond to OA. This is a figure from this study looking at oyster clearance rates at a control group and a high PCO2 group. They have 2 time points, but let's just focus on the first time point for now. So they have a mean clearance rate for their control with some standard deviation, and then a mean clearance rate for their acidified group and a standard deviation. Those are really the main values that you need to calculate an effect size. The formula for an effect size is simply mean of group 1, so that would be this value. From that, you subtract the mean of group 2, which would be this value, and then you divide by the pooled standard deviation. And so the idea is this effect size is this unitless standardized value That could be positive, negative, and in theory, it could be zero, but zero rarely happens. And so then let's say, going back to our, the questions you want to answer, right? You want to say, okay, how do bivalves in general respond to OA? Once we have all our effect sizes, what's next? Well, we calculate what's called a summary effect, which is essentially a weighted average effect size of all the studies that you included in your meta-analysis. Now, what about the more fun questions about addressing some of this between study variability, getting at these local adaptation ideas? For example, you want to see if there's a difference between intertidal and subtidal species. Well, essentially, the idea of what you do is to calculate the summary effect for all the studies that use intertidal species, calculate the summary effect for all the studies that use subtidal species, then you see if you have a significant difference between the two. Very much similar to the structure of a typical t-test in a typical lab experiment. And so in the meta-analysis literature, we call this a moderator analysis, because your predictor variable, your intertidal versus subtidal, is serving as a moderator in your meta-analysis model. And that works fine when you have a categorical factor like subtidal versus intertidal. But what about if you have a continuous factor like exposure time, which is days, a value of days, right? Well, that works how you might think it works. Instead of a t-test structure and an ANOVA structure, you use a regression structure. So in this case, you would try to predict effect sizes as a function of exposure time. So again, just like the structure of a typical linear or nonlinear regression. This is just a different form of moderator analysis here. So now my specific study objectives were to, one, calculate the general outcomes or the summary effects for bivalve metabolic responses to OA. So in other words, we're going to calculate a summary effect for clearance rate and then for respiration rate. And my second objective was to see if experimental design decisions can affect outcomes of individual studies. So we're going to use experimental design decisions as our moderator analyses, or in our moderator analysis. And then my third objective was to see if environmental characteristics, such as upwelling exposure, affect the outcomes of individual studies. And again, this is getting at that idea of local adaptation. And so the methods of meta-analysis are also what you might expect. You simply gather or find all your studies via the World Wide Web. I went to Google Scholar, Web of Science, and located previous meta-analyses to get all the studies that I thought may be included in my meta-analysis. And then once I sifted through all those, made sure they met all my inclusion criteria, and then had to go through each study and pull all the values that I needed for my effect size calculations. So that's the means, the variation metrics, and in my case, the sample size. And it's wonderful when these values are reported directly in the results text or in the table, but sometimes they're in the figures, in which case you use image J to extract them from the figures. Or sometimes those values are so cryptic that you want to just email the author asking for the original raw values. So going back again to our example of oyster clearance rates from that one study at a control pH in an elevated, or a control PCO2 in an elevated PCO2. Let's walk through what the effect size would look like here. So again, mean of group 1 minus the mean of group 2 divided by the pooled standard deviation. And this is important. For my meta-analysis, group 1 in this equation is always the control or the non-acidified group. And then group 2 is always the elevated PCO2 or the acidified group. So in this example here, the mean of group 1 would be 12 because that's the control group, or about 12. And the mean of group 2 would be about 6 because that's the acidified group. And then we divide by the pooled standard deviation. And the important point here is the sign of the effect size is really what you're looking at. 12 minus 6 divided by pooled standard deviation is going to be a positive value, right? So with this setup, a positive effect size means a decrease in clearance rate in response to OA or respiration rate. And again, that's because the mean of group 1 is higher than the mean of group 2. Whereas if this was the other way, let's say our elevated PCO2 had a higher clearance rate, then our effect size would be negative, and therefore that would mean an increase in clearance or respiration rate in response to OA. And again, this is simply because I decided to call group 1 the control group and group 2 the acidified group. So once I had all my effect sizes, I calculated the summary effects of both clearance and respiration rate. And I use sample size as the weights for my calculations. And once you have the summary effect, that's great, but what does it mean? How do you know if it's significant? How do you know if you actually have a consensus response, right? And so what you do is you have your effect size. Let's say again, it's positive. Again, positive effect size means a decrease in clearance or respiration rate in response to OA. Well, that summary effect, or that average effect size, it's going to have some variation associated with it. And so if your 95% confidence intervals do overlap with 0, then you say you don't have a significant summary effect. You don't have a consensus response. But if your 95% confidence intervals do not overlap with 0, then you do have a significant summary effect. So you would say bivalve significantly decreased clearance rate or respiration rate in response to OA. And negative effect size, again, negative effect size means an increase in clearance or respiration rate in response to OA. So yet again, if we have a negative summary effect or negative average effect size, confidence intervals overlap with 0, no significant response there. But if they do not overlap with 0, then we would say that bivalve significantly increased clearance or respiration rate in response to OA. Okay, so now that our summary effects are done, time for, again, what I think is the more fun part, the moderator analyses, getting at objectives 2 and 3. So objective 2 was seeing if, again, experimental design decisions can affect their outcomes. And so the variables I was interested there are exposure time. So how many days were the bivalves exposed to their treatments for? Temperature. Again, temperature was not necessarily a variable of interest in the study, but I simply wanted to see, okay, the temperature that the study was conducted at, did that affect the outcome? And I also wanted to see if the change in carbonate chemistry parameters affect the outcome. So remember, for our surf claims, I talked about ambient pH of 7.7 and acidified pH of 7.3. That represents a drop of 0.4 pH units. So I want to see if that drop, that value, can predict the effect size. And so we didn't just do it for pH, we did it for PCO2 and saturation state, of course. You may be saying, oh, Ray, well, what about total alkalinity and DIC? I very much wanted to do those variables. However, only about half of the studies that I found reported those variables. So the sample size there is probably not robust enough for this type of analysis. And so then my models in this case were mixed effect weighted models. The fixed effects for the predictor variables, so experimental design decisions, and the random effects for genus and study. Keep that in your back pocket for just a moment, but we'll circle back there. And again, my weights for my models were the sample size. And now on to objective 3, looking at those environmental variables. So the first one is upwelling exposure. First, I had to quantify upwelling exposure. And so to do that, I calculated what's called Bakkun's upwelling index, which is essentially a measure of Ekman transport, either on or offshore. And as Bakkun's upwelling index, it's a uniless value from about -250 to about +250, where a more negative value represents strong downwelling, and a more positive value represents strong upwelling. And a value of 0 would mean neither upwelling nor downwelling. So you have very little across shore Ekman transport. Or rather, or maybe they just average each other out in the opposite directions. And so these are what my upwelling index values look like across the globe for the studies that I analyzed. And for those of you who are familiar with the upwelling regions, this is kind of what we would expect. So for example, the Peru-Chile coast, one of the strongest upwelling regions in the world. That's where we had our highest values. Coast of Asia, not much upwelling, more downwelling over there. Our upwelling index values matched that. Europe, a mixed bag, depending on the angle of the coastline. And I'm not the most familiar with local oceanography in Maine, but I know there is some upwelling here occasionally, and that's what these light blue values, or not, circles here suggest. So my point here is these upwelling index values that I used in my models seem to accurately represent quantifying upwelling exposure throughout the world. And then for our categorical analyses, intertidal versus subtidal, and infaunal versus epifaunal, we saw if the Q statistic was significant. And again, mixed effects, weighted models. Let's talk about this for a moment, though. Why did I include genus and study as a random factor? So Many studies have many effect sizes, more than one effect size. For example, if a study had 3 pH treatments instead of 2, you're going to have an effect size for every pairwise combo of treatments. You're going to wind up with multiple effect sizes there. If the study measured clearance or respiration rate at multiple time points, like that example I showed you, you're going to have a suite of effect sizes for each of those time points. Or if the study looked at more than one species, you're going to have multiple effect sizes per species. And so, I didn't want to just pick one of those effect sizes, right? That would be kind of arbitrary and you'd lose information there. So instead, I included them all, but I identified study as a random factor to account for the non-independence of the data points within a study. I also included genus as a random factor to account for the fact that species that are more closely related may be semi-independent from one another compared to species that are more distant from one another. Okay, now into the results. First, the summary effects. So what you're looking at here is called an orchid plot. We have positive effect sizes and then negative effect sizes. And each of these circles or each of these bubbles is one effect size. The size of the circle or the bubble is determined by the weight, which is determined by the sample size. So you can see we have 203 effect sizes for clearance rate coming from 31 different studies. And so this circle right in the middle here with the black outline, this is the summary effect. The black bands around the circle are the 95% confidence intervals, and the thin lines are the prediction intervals. So we can see our summary effect is 1.38. And we can see that our confidence intervals do not overlap at 0. So we do indeed have a significant summary effect. More specifically, we would conclude that in general, bivalves decrease clearance rate in response to OA. Again, remember, positive effect size means a decrease in clearance or respiration rate. And the summary effect for respiration rate, very similar to clearance rate, just 0.1 units lower, but again, significant because their confidence intervals do not overlap with 0. Okay, let's take this figure moment by moment. So here we have a linear model predicting effect sizes from upwelling index values. Remember, a highly negative upwelling index value means strong downwelling. A highly positive upwelling index value, strong upwelling, and then an upwelling index value of 0 means neither upwelling or downwelling dominates. And positive effect sizes, which I highlighted as red here, represent a decrease in clearance rate in response to OA. The negative effect sizes represent an increase in clearance rate in response to OA. So again, the different bubbles are the different circles. The size, the different bubbles are the different studies. The size of the bubble is determined by the sample size of the study, and the different colors come from different studies. So the same color means the effect size came from the same study. But we can see we have a highly significant negative relationship here with low upwelling index values or high downwelling areas leading to higher effect sizes. So in other words, when we have strong downwelling, we have bivalves that tend to have strong drops in clearance rate in response to OA. Whereas when we have strong upwelling, we have bivalves that either don't change clearance rate in response to OA or increase clearance rate in response to OA. Now for respiration rate, we see a similar but slightly weaker trend where again, strong downwelling, strong drop in respiration rate. Strong upwelling, less of a drop, if not no drop in respiration rate in response to OA. Now going back to orchid plots for our other environmental variables. So here we're looking at the summary effect of subtidal species and intertidal species for clearance rate. But we can see we have no significant difference between the two. However, for clearance rate, we do have a significant difference between the two. The summary effect for respiration rate for subtidal species is significantly lower than that of intertidal species. More specifically, we can see that intertidal species significantly decrease respiration rate in response to OA. Whereas for subtidal species, because these confidence intervals do not overlap with 0, we would say subtidal species either don't change respiration rate in response to OA, maybe they increase it a tiny bit, or maybe there's just too much variability and there is no consensus response. But the main takeaway here is there is a difference with intertidal species appearing to be more sensitive. The substrate type, epifaunal versus infaunal. no significant difference there for clearance rate. And yet again, no significant difference for respiration rate for epifaunal species versus infaunal species. So now moving on to the experimental design decisions. So temperature, we see a significant positive relationship where studies with low temperatures tended to have either increases in clearance rate or no change in clearance rate, whereas studies with high temperatures tended to have a bigger drop in clearance rate in response to OA. For respiration rate, slightly weaker relationship, but still significant and still positive, with bigger temperatures leading to bigger drops in respiration rate. Now, exposure time. So exposure time, again, is the amount of days the animals were exposed to their treatment for. For clearance rate, we see no significant difference, or no significant relationship, rather. However, for respiration rate, we do see a slightly negative relationship here, with studies using low exposure times having a bigger drop in respiration rate, and studies using high exposure times having a smaller drop in respiration rate. Now on to the changes in the carbonate chemistry parameters. First, we're predicting clearance rate effect sizes as a function of change in saturation state. And we can see that we have no significant relationship there for clearance rate. But for respiration rate, we do see a significant relationship. More specifically, we see bigger drops in saturation state lead to bigger drops in respiration rate, which is what one might expect. And for pH, we see the same thing. Bigger drop in pH means bigger drop in clearance rate in response to OA. Respiration rate and pH, the same thing. Bigger drop in pH leads to a bigger drop in respiration rate. Now, for PCO2, the x-axis has negative variables, or rather negative values. This is again because it's what I called group 1 and group 2. And the saturation state in pH, that gets lower with acidification, but PCO2 gets higher with acidification, right? That's the only reason these values are negative. But the idea here is the same. A bigger change in PCO2 leads to a higher effect size, which means a bigger drop in clearance rate. And same thing for respiration rate. Bigger change in PCO2 leads to a bigger drop in respiration. Okay, now on to my very brief discussions. I probably have not too much time left. Regarding our summary effects and our broad conclusions, we saw significant summary effects for clearance and respiration rate. So we can say that in general, bivalves decreased clearance and respiration rate in response to OA. This is what you would expect. This makes sense, right? You know, OA is stressful for the animals. Stressful conditions lead to depressed metabolic activity. So what we're seeing is probably just a stress response. And at least on the clearance rate end of things, this may provide mechanistic insight for changes in growth. Bivalves often decrease growth in response to OA. Well, maybe it's because they are taking in less food, because we showed that they have decreased clearance rates in response to OA. Now on to tidal exposure. So subtidal versus intertidal species. This was an unexpected conclusion. We saw no difference for clearance rate, but we did see a difference for respiration rate. More specifically, we saw that subtidal species either increase or don't change respiration rate, but intertidal species decrease respiration rate. So it seems as though the subtidal species may be a little less sensitive and the intertidal species may be a little more sensitive. This is suspicious to me. First of all, I think it's suspicious that we have different outcomes for the two different responses. However, that could be because of a low sample size. So a lot of my studies that I found from my meta-analysis were oysters and mussels, which can live in the intertidal. So we didn't have many strictly subtidal species. So the sample size for the strictly subtidal species was small. Perhaps that's leading to these odd results. However, this difference is also the opposite of what I would have expected. I would have expected the intertidal species to be a little more resilient to ocean acidification than the subtidal species. Now, regarding substrate type, we saw no difference here. Infaunal species didn't appear to be more or less sensitive than epifaunal species. This, to me, again, was surprising. Yeah, so as Rob showed a couple of weeks ago, poor water chemistry in Maine That can lead to pHs below 7.0. So I would have thought, okay, these infaunal animals that are spending time in these super harsh conditions, because they're used to these naturally acidified conditions, they're going to be more resilient to ocean acidification, as opposed to epifaunal organisms, which don't deal with that harsh pore water chemistry. But my meta-analysis showed no difference there. I'm not too sure what's going on. Perhaps it's because while the shell material of the animal is interacting with the pore water chemistry, the tissue and the organs of the animal are not interacting with the pore water chemistry, especially with the siphons coming up top. Just kind of a shot in the dark there, but that could explain the lack of difference or the lack of pore water chemistry effects. And so while tidal exposure and substrate type didn't do what I wanted them to do or what I expected, my upwelling exposure relationships were exactly what I hoped. More specifically, my results suggest that bivalves from strong upwelling areas are either more resistant, more resilient, less sensitive, whichever of those phrases you think works best, to ocean acidification. And this aligns with a couple of previous studies that have kind of got the same idea. So a recent meta-analysis showed that animals from environments with high PCO2 variability tend to be a little more resilient, a little more resistant to anthropogenic ocean acidification. These upwelling results get that same idea, but just focus on upwelling as a mechanism to affect natural carbonate chemistry. But I think this raises some cool questions or future directions. For example, is this apparent resistance or resilience a plastic response or a genetic response? And also, this is probably getting way ahead of myself, but this may present an opportunity for selective breeding where these more strong bivalves in these upwelling areas can be used in aquaculture efforts. Now, for my experimental design decisions, the outcomes I saw here aren't too surprising, what you would expect. And I want to highlight, I performed these experimental design analyses not to inform decision making. Ultimately, if you're conducting a new experiment, the context of your system, your environment, your species, and your question should dictate, your change in carbonate chemistry parameters, your temperature, your exposure time. So I'm not trying to inform decisions there. I perform these analyses simply to provide context for outcome interpretation for additional studies. So let's say, you perform a new study and you expose your bivalves to a very low change in pH, a little below 0.2 units. But you find a very high a very high effect size. So your data point, your outcome would be kind of an outlier here and would suggest that your species, your population, your study was surprisingly sensitive. So that's all that these results are here for, just to provide context for additional studies. And with that, I will take questions and even comments too. Again, I very much encourage comments.
Thank you, Ray. That was a really informative presentation. And if anyone has a question, you can raise your hand or put it in the chat. Carolina.
Hi, Raymond. Thank you so much for your presentation. I wonder, I would like to, you probably said this, but I missed it. Like, what is, you're only focusing in bivalves as your species for this meta-analysis, or did you include any other mollusk, type of mollusk?
Yeah, so I only included bivalves. I've toyed with the idea of doing other mollusks, or even crustaceans or echinoderms, for the sake of a stronger publication. But ultimately, it's going to require a lot more time. But if my colleagues or reviewers say we want more than bivalves, that may be the endpoint here, is to look at other taxa, other phyla as well.
All right, thank you. And I guess that my other question will be, can you elaborate a bit on why? I'm surprised on your expectation of more resilient or more yes, more resilient individual or species from the intertidal. So I mean, these are animals, these organisms are really on the verge of many thresholds in their environmental conditions that they are. So why is your expectation of higher resilience for the intertidal ones?
Yeah, so I've been putting a lot of thought to this question recently. My original idea was, okay, intertidal organisms, they're used to a very harsh environment. Sure, they're not underwater for their amount of time as subtidal species, but they have to deal with air exposure and higher temperatures because they're intertidal. And so I guess I was trying to say, or my idea was, while ocean acidification stress is different than that air exposure stress, it's still a very harsh stress. And so these organisms may be adapted to just deal with harsh stress in general. But as you were probably alluding to, maybe the fact that air exposure stress is so different than ocean acidification stress that there's just no link between them. And maybe that's what's going on here. Sure, they're used to stress, but they're not used to OH stress.
All right. Well, thank you so much for your answers and presentation again.
We have a question in the conference room at DEI.
Hey Ray, it's Rob. Fantastic presentation. I was just wondering, as you were doing your lib review for this project, were you able to get most of the data just by reading the papers? Or did you have to reach out to the authors? And if so, did they generally play ball and send you what you wanted?
Yeah, so I had 31 studies for clearance rate. I think it was 26 studies for respiration rate, accounting for some studies that didn't overlap in responses. It's close to about 40 studies. And of those 40 studies, I'd say, I think 35 of them, I have the values somewhere, 35 of them, I was able to pull the data directly from the papers. So a few of them I did have to email. I think all but one responded to me. So most of them played ball. But what was pretty cool is when I would email authors, they would often say, oh, wait, we also have these data, which we didn't publish, but, and we're not going to publish. So you can use those in your meta-analyses. So for example, one time I contacted an author to get clearance rate data. They're like, oh, we actually have the respiration rate data, but we didn't publish it because we didn't find a significant response. I'm sure it's not significant response, so reviewers may not like it, but ultimately that matters for my meta-analysis. So I would get to use those data. Or sometimes an author would say they only reported data for one species because the other species they didn't find a significant response. I got my hands on those data. There was one time where an author wanted co-authorship for providing the data, and I said thanks, but no thanks. But for the most part, they played ball with one or two exceptions.
Okay, great. And you had shown me one paper that you were reading that had suspiciously small error bars. Were you able to resolve that either by reading the paper or receiving the data?
Yeah, I'm trying to remember that particularly, or that predicament. I think I wound up just, trusting that there was no error there. Because I think I looked at other papers that author did, and they also found small error bars. And I forget where our exploration led us to there, but I ultimately just wound up trusting what went through the peer review process there.
Okay, great. Thanks.
John?
Hi, Ray. Thank you very much for the presentation. I enjoyed it very much. I'm interested in the upwelling question. I mean, I never would have thought to look at areas where there's upwelling versus areas where there's downwelling or no real change in water being brought up from depth. Was that your idea, or perhaps I should ask how much of that was your idea?
Yeah, so thank you for that question. I'm excited to actually kind of brag about this a little bit. So one of the core courses at Stony Brook had us conduct a review paper. The topic I chose for my qualitative review paper was, ocean acidification, bivalve, metabolic responses. And eventually what I did is I went up just making a table showing, okay, which studies, showed increases, decreases, and no change. And at the end of my table, I was looking, and I was like, okay, a lot of these studies that didn't have changes in clearance rate or increases in clearance rate came from, either looked at mussels or came from the Chile, Peru coast. And then the semester before, taking oceanography courses where I learned that coast had high upwelling. And so, I mean, it may have been implanted in me before I did the study, but I like to think that hypothesis was mostly my own. That's why I was excited to see that I did find a significant relationship.
Right. Yeah, I think that's very, very interesting. And I hope you pursue it, and I hope that you can find differences at the field level over these broad geographic areas. That would be quite an advance. I have a more mundane question, and that is the sources of the individual organisms used in each of the experiments and the publications you reviewed. How confident are you in, let's say, subtitle versus intertidal?
Yeah, so if the authors didn't—
How confident were you?
Is my connection dead or?
No, I'm okay now. It was dead for a second.
Assuming I interpreted your question correctly.
Yeah.
So my approach here was, okay, a lot of the authors, I'd say over half of them specified where their organisms were collected from, intertidal zone, subtidal zone. However, sometimes the authors didn't specify, or sometimes they came from even an aquaculture facility. And so when that was the case, I went into the literature to see, okay, is that species predominantly, or is that species commonly found in the intertidal zone? Regardless of where that specific population was, is that species commonly found in the intertidal zone? And if the answer was yes, I grouped that as intertidal.
Okay.
If the answer was no, like for surf clams, I put that in the subtidal.
Yeah. Okay, great. Thank you very much.
Are there other questions for Ray? So Ray, you said your next steps is to create the models for your dissertation using some of this information.
So the next, the study I presented today, it's basically done. I'm reworking the paragraphs for the discussion, but the quantitative aspects of the study, I'm happy with, and I'm based off what I learned about meta-analysis. I think I did everything robustly enough to meet the peer review standards. But regarding other modeling, other work I have to do, that's going to be using the results from the lab experiments I conducted this summer, which don't pertain to ocean acidification at all. It's more of the food availability aspect that you had mentioned in your introduction. But yeah, so maybe next summer at the seminar series, I'll be able to present those results.
Great. Well, if there's no other questions for Ray, we'll wrap up. And I just want to thank Ray for his presentation. It was really interesting. I learned a lot about meta-analysis, how to conduct one. And so thanks everyone for joining us this afternoon.